Hi kids, so let us now continue with our discussion. So for the second part of your video lecture, this is all about the steps in your automated analysis. So in terms of the different steps, so we will be focusing on each phases or each steps in how we analyze your different substances katong gikan pa sa pre-analytic phase to analytic phase to post-analytic phase. Na kumbaga, each na mga substeps ana na phases, i-discuss nato siya in terms of unsa na mga portion, di ba? What portion or what things can be automated in that part. And for this slide, as you can see here, you have table 7-2, which is already a summary of chemistry analyzer operations. And ito po, kani siya, na makita ninyo diri kanina part, monito siya ang ako ang ginamin na taga-step sa inyuhang automated analysis. Dalang po tayo ipang explain gamay na unsa ang mga automation or unsa ang mga improvement na gipang buhat para ma-automated ni sila ng mga parts. So, makita man ninyo sa inyong handout, dili lang po na ko na siya isa-isahon since as we go along our lecture, ako lang po din siyang ma-discuss isa-isa. So, let's proceed with our first step, which is your specimen preparation and identification. Now, as you can see here in this table, these are different technologies used for automatic identification and data collection of your specimen. And actually, some laboratories are already using this one, especially when we talk about barcoding. During my internship, isa sa akong nahibalaan na naga-barcoding is ang SPMC. I think Davao Doc also, pero since during my time, wala man ko naka-internship, wala pa, dili pa affiliated sa SPC with Davao Doctors. And even katong nag-clinical, nag-CI, wala po ko na-assign pa sa Davao Doc. But then, it was mentioned that they are also using this type barcoding. And actually, barcoding is good. Kay, kumbaga, mura magugmahan ay tanan processes like there is lesser or minimum na problems in terms of labeling errors. Kay since encoded naman siya daan sa computer, even ang mga test na i-undergone, there is minimum kaayo na chances na naamoy test na ma-mislook. Kaya again, everything is encoded. All you need to do is just to scan that barcode and then let the computer or let the auto analyzers do its job. Now, there are a lot other than your barcoding. As you can see here, we have your optical character recognition, voice identification, social na kaayo, touch screens, of course, common, touch screen naman yun ang kadaghanan nato ng mga computers attached to your auto analyzers. Pindot-pindot na lang dayon. Then you have your light pens, your radio frequency identification, your optical mark readers, and your smart cards. Now, uh, if you could still recall during your PMLS2, 'di ba isa biya sa pinaka dako kaayo og problem is kanig yung specimen identification. That is why there is proper labeling and such. And actually, sa tanan mga steps, isa po din siya sa medyo lisod yun kaayo i-automate. But then again, daghan man og ways, and these are just some of those examples already. So na ako'y mga images diri, these are just examples, 'di ba? At least na ma-picture out ninyo unsa tong mga technologies na gi-mention a while ago. So this is your optical character recognition na by the use of a pen na ay software, na ay software na i-install sa computer, and then with optical character meaning kana mga ginasulat, handwritten, iagi lang na siya sa pen and then ma-recognize sa dayon na nila. So, na siya. And then we have kani, MICR, this is your magnetic identification character recognition. And MICR is actually very common sa mga check. So you can see here ka mga check sa kwarta na labi na ang kanina encoding na number part. So this is actually magnetic. Mo na siya ang gina-refer sa magnetic identification character recognition na naada a specific na scanner for those checks for double checking if ano po jud, if legit jud siya na check, and then kana na siya ang ginabasa as you scan the check. And then this one, This is your optical marker recognition. And the best example for this one is actually your Scantron, na 'di ba? Kaya siya optical marker kasi we're just placing marks just like your Scantron na shade lang mo sa inyong letter so kana na siya ang gina-recognize pud ani sa inyuhang optical marker recognition. So other emerging technologies as of the moment is one is your automated specimen inspection. Actually, while I was reading the definition of this, I guess this is found in your Henrys. Medyo na-amaze ko. And when I tried to search for videos about this one, daghan na po jud og mga machines na naaiing ani na klase of automation, your automated specimen inspection. So what is this all about? It is for identify sample identification errors and even your sample integrity issues. So unsa man ning mga common sample integrity issues? Like for example, hemolyze inyong sample mo magyud ang number one. So with automated specimen inspection, it is able to check or verify na inyong sample di ay hemolyzed. Unsa pa man ang mga problems, lipemic, icteric, so kana, they can actually, or kana mismo na machine ninyo, they can already check kung unsa kagwapo ang integrity sa inyuhang sample. It could also sort a random collection of different size containers with different additives. Ah, Siyempre nakakita naman gyud mo og different na mga tubes, 'di ba? Naik uban na capacity of up to 5 ml, katong mga tag-as kaayo, and then you have your katong mga normal tubes na atong ginagamit, and then naata atong inyuhang mga for capillary na 'di ba mga gagmay mga capillary tubes. Now, in a machine, whenever you place different size na tubes, actually ginasort, kita mismo ang nagasort. And gina-input nato na siya properly pod sa system sa computer na kunwari kani si sample number 9 butang ninyo sa rack, small na tube ni siya and all. But with automated specimen inspection, especially if you have a total laboratory automation na system gyud sa inyuhang laboratory na pati pag-sort out sa specimen, kanina siya ang ginamina. Even pag-sorting, kay kabalo na ang inyuhang auto-analyzer na kaning gamay, ato ni siyang ibutang tanan gamay diri sa isa ka rack, tanan na kaning tanan na red top, tanan na lavender top, and such. And then assess each sample for proper labeling, adequate volume and interference from icterus, lipemia, or hemolysis. So ingani kanindot ng mga emerging technologies as of the moment. So, syempre if you will ask me if na ako'y na-encounter na ani na machine, wala pa. Kumbaga mata-meter gyud na ani na mata-matahon gyud nato na hemolyze ba gyud ning sample, needs repeat collection and such. Now, another emerging technology is, and na-mention po ni siya kaganina dito sa inyuhang box, your radio frequency identification or your RFID. Now, I guess you are familiar with this one just by looking at these images. Common kayo ni siya na ginabutan sa mga tag, sa mga clothes, na kumbaga, pag, di ba, pag mugawas mo niya, dili ni ma-remove ang RFID, kay butingog yun ni siya, something like that. So, ingana pud na ang RFID diri sa inyuhang diri sa ato ang mga auto-analyzer. So, passive RFID is passive and without active human intervention. Kaya 'di ba, ibutang lang gyud nato na siya dira as part sa label sa inyuhang sample, pwede dira nato ibutang sa tube. So, mura siya og barcoding but different na kanang style. So, the advantages of your RFID, no line of sight reading, dynamic data storage, and it is not affected by cold storage. Kaning manggong RFID ninyo, maski na daw na kanang medyo matabunan lang gamay kay walay problema kay it does not need as mentioned, it does not need kana line of sight reading na dapat yun matutok nato ang scanner dito sa iyaha. So kana lang pud ang nakanindot sa iyaha na even if napa na siya sa sulod sa box or what kay ma-scan gihapon siya. The disadvantage here is that it is yet to be commercialized on a large scale in the clinical laboratory, and syempre, as always, mao gyud ni ang common na disadvantage gyud, greater gyud ang cost. Bahal daw ning mga RFIDs. So this is just a short video para naamoy ma-picture out as to how di ay pwede ma-automate ang kaning in terms of your sample identification. So kindly watch this video. So as you can see, Beckman Coulter is actually also a very famous na brand of your auto analyzers. And as you can see in the video, di ba, na nakita dito 3 seconds specimen checking, and it can even sort out different sizes of your tubes. Tapos naana na siya track or naana na siya conveyor na siya na bahala kung asa padulong ang inyong sample, kung asa siya na section. So ingana ka kuyaw ang mga total laboratory automation gyud kay sila man gyud ni ang kanang naa conveyor na that could easily send samples to different sections in the laboratory. Now we proceed to your specimen delivery. So kani uh, naa na pud tay mga ing-ani na automation here in Davao also. So number one is your courier service. Dati, dili common ning courier service, especially in terms of sending your different samples, especially when it needs confirmatory testing. As far as I could remember, dati ikaw personally, it's either ikaw personally magdala sa imong sample or dito jud ka magpa-test, ikaw mismo ang moadto dito. And usually ang mga confirmatory testing centers, di ba naa sa Manila. Imagine unsa ka gasto. Now, as of this time around, ang hospital na ay ang hospital na ang pwede na magpadala sa kung asa ang confirmatory testing center. So nalay additional na payment, ibayad ang patient ana, but at least mas barato and mas less ang hassle. Next is your pneumatic tube systems. Now, pneumatic tube, I guess, napansin siguro ni ninyo kung nakaadto na mo sa SPMC also and in Davao Doctors. But so far, sa ako ang nahiringan, dili kaayo ginagamit si pneumatic tube system sa SPMC and sa Davao Doc kay somehow namang gud tay tan-awon ani na medyo na ay problem or disadvantage with the use of your pneumatic tube systems and mao ni siya ang ako ang i-present. So one, mechanical problems in the switching process have been known to cause misrouting of carriers. Imagine there are a lot of sections or departments in a hospital. 'Di ba? Kunwari from fourth floor, kung pila man naka-floors ang hospital, and then nakabay ano pag yun na kung ward ba, kung private ba, and then sending of specimens, tas kunwari daghan mo mag-send. So, not really possible na 'di ba na ma-misroute ang kato na mga sample. And then another, it is very prone to hemolysis. Why? Because of this one, 'di ba naay times na naay sudden acceleration and deceleration na mahitabo as you transport your samples through your pneumatic tube systems. And prone to hemolysis because it might depend on how the personnel or the person handling that specimen properly pack that material na dako gani pwede, dili man sa dako, pero there are really chances na pwede, di ba na, pwede siya mabuak, pwede siya na, basta in terms of specimen integrity, somehow it could affect. And actually, this is one common study na ginatanaw na unsa ba jud ang effect sa mga samples if it is delivered through pneumatic tube systems. Actually, pwede na siya ninyo studyhan. So this is just a short video about or what a pneumatic tube system looks like. If in case na wala pa mo kakita, or if nakakita naman, then you can just skip this video. Okay, so I hope na-appreciate ninyo siya. If you are going to ask me if I have actually seen or used a pneumatic tube system, sorry kids, wala pa kay. During my internship, wala pa siya sa SPMC. And yes, nga na nagyud ko katigulang, di ba? Wala na nako siya naabtan. And even during my katong CI days, nako kay wala man po jud ko na-assign. So as in, totally wala po jud ko nakakita. So other specimen delivery pa na type, you have your electric track vehicles. Now, these are larger carrying capacity compared to your pneumatic tube system, and it is not associated with problems such as damage to your specimens caused by acceleration and or deceleration forces. So later, nalang po ko yung vid na ipakita ani. Let's proceed to the next. We have your mobile robots. Now, mobile robots, delivery of specimens to lab benches by a mobile robot is usually more frequent than human pickup and has been shown to be cost-effective. Mobile robots, literal na murag, instead na ikaw personally mag-deliver from one department to the other, na atay, mura siya og kanang cart, mura siya og kanang inyuhagong carts na instead na kamo personally na magdalaan na cart, transferring one siguro unsa one thing to the other, kanina na siya inyuhang mobile robots. So para mas ma-imagine ninyo, I also have a video for that. So let's proceed to the next slide. So the first video, this one, is referring to your electric track vehicle. Now, grabe ni ka-cutie actually kay kani siya na electric track vehicle is in a nursery or kanang children's hospital. So kindly watch this video. Wala gyud ni siya sounds kids ha. Okay, so let's proceed to this one. Di ba nakita ninyo? If we compare electric track vehicle to your pneumatic tube system, it really has a larger capacity. And instead of having problems with deceleration and acceleration, actually kani smooth lang kay naa naman gud siya iyahang kumbaga mura lang gud ka nang sa train na siya speed na daan na nakaset and dili kayo necessary na kanang ikaw pa ang kanang mo kuan sa iyahang speed, just like that. I hope you get me. Next is katong sa inyuhang motor robot. O 'di ba, you see what I mean, na mura lang siya og kanang mga cart, pero instead of someone having to push or pull that cart, automatic na siya na lang gyud mag-go from somewhere to another, transport certain items. So ingana na inyuhang mobile robots. So let's proceed to the next step. Your dili pa din ni siya next step. Rather, sorry. So, preparation of your sample for analysis has been and remains a manual process in most laboratories, which is true. Actually, kani magong specimen preparation and identification, if tan-awon jud ninyo medyo lisod man gyud kay ni siya i-automate, and if gusto nato siya i-automate, then that is an additional na cost na pod. So some alternatives for specimen preparation and identification with the use of robotics or front-end automation, as what has been presented a while ago. Bypass specimen preparation by using whole blood for analysis. Kasi syempre, if we need to use a sample, a serum sample, di ba remember, it takes a lot of time to get that sample since ipa-properly clot pa ninyo ang blood and then i-centrifuge pa ninyo siya. Tapos di ba para ma-properly clot yun din yung sample, it takes about 15 to 20 minutes. And then in the centrifugation process, it takes about another 15 minutes. So dugay good guys siya. And time is of the essence in our work. So, in order na ma-lessen or ma-cut off na siya na time, then it would be best to use a whole blood analysis. But naaman po tayo pros and cons with the use of your whole blood. Kaya naaman po tayo uban substances sa ato ang body na mas gwapo or mas okay ang iyahang levels kung dito mismo nato siya sa serum, kay mas present man siya dito sa serum compared to your whole blood. Though I could point out that there are really auto-analyzers as of the moment na naingani na ang ilahang ginatanaw na instead of using the serum, then we will be using whole blood. Like, for example, uh, instead of using serum or plasma, rather, just like your, say tawag ani, sa ABG, your arterial blood gas analysis, na ay mga samples or naming machine once na murag ipa-check sa amoa na whole blood ang amo ang gamito na. So kana, and even ang kato sa inyuha mga CRP na usually mga serum sample, ang ginapagamit na ana is sa mga machines na murag set up siya na whole blood na ang gamiton, kay it is actually very much easier if we will use that. But then again, as mentioned, natay pros and cons. So use of plasma separator tube and perform primary tube sampling with heparin in plasma. Now, what is the reason for this? It actually eliminates the need both to wait for the sample to clot and to aliquot the sample. So, as explained a while ago, dire-diretso, labi na kung kani sila. And then, lastly, barcode labeled tubes. So, as you can see here, the mga images sa inyuhang right side, even kanina part, this is actually an example of your auto-analyzer na siya na mismo ang mag-aliquot sa inyuhang mga samples. You just have to place your sample and and another test tube where the aliquot will be placed, and then just set gamay na setup kung pila ang volume na needed for transferring the aliquot. So, kanina siya. And then, of course, your robotics with barcode labeled tubes na agad-agad, scanning na lang, and then transport na dayon sa inyuhang mga samples to its designated sections. So now we go to your next step, your specimen loading and aspiration. Now, circular carousels or rectangular racks are used in a auto-analyzer as your specimen containers. So I have pictures for this one. Depende siya sa design sa inyong machine that they could either use a circular carousel or a rectangular rack. Trays or rocks are numbered to aid in sample identification and move automatically in one position step at pre-selected speeds. Even in your trays, there are barcodes placed and mga numbered barcodes so that when you set or encode ang kaniinyong mga samples in the computer of that auto-analyzer, inyo siyang iset up inline sa kung unsa pud ang katong barcode sa inyong mga racks para na ay proper identification gihapon sa inyong mga specimens by the time na mo load na siya pasulod sa inyong machines. And then instrument can determine the slot number containing the last sample and terminate the analysis of after that sample. So yes, ingani ang inyong mga auto-analyzers na by the time that it will reach at the last specimen that you've encoded, then the whole processing will just stop or halt. It can actually determine that. Then instrument's microprocessor holds the number of samples in memory and aspirates only in positions containing the samples. So, if in case na, just like for example, kunwari na amoy rock holder that can hold up to 5 samples, and then you only place 2, and then empty na ang 3 slots, your auto analyzers could actually also identify na duha lang yun dito sa inyuhang uh, rock ang naa lang jud sample. So again, it's a matter of encoding and scanning. Other than encoding, scanning of your machines sa kung unsa ang naagyo dito sa inyuhang rock. So these are just some examples of your racks as mentioned. So this is your circular carousel. And actually, nanay mga designated numbers lang po na inyo makita. And then this is your rectangular rocks. And this is how you itsura sa loading zone sa inyuhang auto analyzers. So as you can see, diba, there are empty slots on some of the rocks. So again, wala tayong problema with that. Your machines could actually identify that that is empty. So actual measurement of each aliquot for each test must be very accurate and is generally done through aspiration of the sample into a probe. Now, we do not have any problems in terms of the volume of your sample kung pila ang i-aspirate sa inyong mga machine kaya that is programmed already. And there are actually some, like for example, tubes na gyud ang inyong ibutang sa inyong rack. There are some machines na naana sila ginabutang na designation na dapat ang inyuhang serum is up to the jud siya na level. Kaya if it will not reach to that level, then you need to transfer it to a smaller containers. Mga ingana. So natay mga machine na ingana na naapod na sila limit na although kung dili man pre-measured, naapod na sila ways na ma-identify na up to diri na level ang inyuhang volume lang volume sa inyong serum na naa sa tube. So tubes are typically decapped before sampling, but there are machines that offer cap piercing technology. Sa katong nakita ninyo na video kaganina, di ba naatoy isa dito na naatay mga auto-analyzers na sila na mismo ang nagadika. But for some, and unsa ang most common na auto-analyzers here in Davao, especially here in Davao, kita mismo nagadikap ana. Mo na medyo, since kita kay modular integrated system lang man po jud ta, isa na sa murag hazard na ginabuhat sa chem na syempre pag magdikap biya ta there could be aerosols or there could be spilling. Sometimes sabi na kung gahi jud kayo tanggalon ang inyong cup, then there could be spilling of your sample. So careful lang punta ana. But there are machines na no need for you to decap. Siya na bahala mo ni offer cap piercing technology. So an example for that is your Unicell analyzer, which is from your Beckman Coulter. And isa sa best example na ako mahatag na ani, other than your Unicell analyzer that has a cap piercing technology, that would be your hematology analyzers. Labi na katong mga Sysmex and Pentra na brand sa machines na ako ang nagamit, na encounter na ang style ana is we do not have to remove the cap, just place it automatically in your racks. Isulod lang po dayon sa inyong machine and then wala na, siya na bahala i-pierce na ana siya piercing technology na siya na magkuha og blood. There are also machines na you do not need also actually to mix your Siyempre, upon collection, mix gyud na kadali. But by the time that you will process it in the machine, dili ninyo kailangan na i-mix og usab kay there are machines na sila na pud mismo ang naga-mix og balik sa inyong mga samples. Now, uh, the commonly used wash solution, or actually ang wash solution gyud na ginagamit is your deionized water, and sometimes it is with a surfactant. Wash solution is important and dapat dili ni siya mabutangan og other pa na mga reagent kay since this is necessary, labi na kay 'di ba probes biya na ang naa, and ang mga probes biya ninyo dili man ni siya na taga isa ka sample na ay isa ka specific na probe. Every after ang kaning probe ninyo mukuha og sample sa isa ka patient, iha man ang hugasan sa before na mo proceed na pud siya og kuha sa another sample sa next ninyo na patient, sa next ninyo na tube. So mo na naagid ning wash solution dapat kay this is actually for cleaning your especially your probes to prevent and minimize carryover problems. So, mao na siya. Ang common man gyud nato na problem diri in the machines are carryover. Na it may carry over your kung sa man na inyong sample from one patient to the other, and that may lessen accuracy of our results. So that is why there is a need of cleaning gyud. Cleaning every now and then sa inyong machines, pero in terms of cleaning your probe, automatic na gyud na siya na part sa processing sa inyong machine. So we have this what we call your loading zone, and I guess na-mention ako ni siya. Now, loading zone is the area in which specimens are held inside the instrument before they are being analyzed. Now we go to your specimen processing. Now, autoanalyzer should be capable of removing protein and other interference. Now, how to remove protein and other interference? It could be through dialysis, column chromatography, and filtration. So I think you all know, you are already familiar with these three. Diba, dialysis, process of removing excess water, solutes, or toxins in our body. So, murag ing-ana po iyahang thought in terms of removing your protein, your column chromatography, and then your filtration. Now, you might wonder why need i-remove si protein during the processing or other interference pa. Protein is a very common interferent. It could, one problem man gud na gina-post ni protein is, number one, ang iyahang size. Na kumbaga, common kay isa na mo block. No, i-block niya ang other substances because of its size. Another reason in terms of our auto-analyzer, siya ni ang common pud kaayo na makakause og blockage sa inyong mga tubes, sa inyong mga probes, sa inyong mga tubings, dara sa inyong analyzer. Labi na kung dili mo mahilig gyud kanang mag-cleaning first thing in the morning before you use your machine, this could really cause a problem. Kay mura man gud na siya matrap sa side sa inyong mga tubes. And syempre, as we all know, protein is already a big sized na molecule. So that is why cleaning gyud mo, and as much as possible, your auto-analyzer could remove na a certain process siya na buhaton to remove proteins. Now, may mga test actually, I don't know if na-point out siya sa inyuhang lab, but if napansin ninyo sa inyuhang mga package inserts, di ba namoy mga pre-treatment po dyan na ginabuhat sa inyuhang mga samples? And why you do pre-treatment, isa po ni siya ka-reason, di ba? Removing of interference na naasa mo'y mga buhaton na processes, na ba mo'y i-add na reagent just to remove this interference. Or, pwede gani na to i-example during kanang inyuhang call this one kana sa inyuhang blood banking, na kana mag-cross-matching mo na jud na 3 times washing sa inyuhang blood, kani po na siya to remove protein and other interferences. Next is your reagent systems and delivery. Now, in terms of your reagent system and reagent delivery, we have two types. You have your open and closed system analyzer. So what are these two? So for your open system analyzer, operator is able to change the parameters related to an analysis and prepare in-house reagents or use reagents from a variety of suppliers. Your open system analyzer is very flexible and adapt readily to new methods and analytes. How about your closed system? Closed system requires the reagent to be in a unique container or format provided by the manufacturer. So to explain further, from the word open system, meaning class, ang hardware, software, ang instrumentation, it comes from different na mga manufacturers. Na kumbaga muragi tigpi-tigpi ninyo to put up a one auto analyzer. And kaning ginamin sa in-house reagent, it does not necessarily comes from asa ninyo napalit ang kanina specific na manufacturer. Pwede ka mo mismo magbuhat, di ba mo man ang ginamin sa in-house, pwede ka mo mismo ang magbuhat and then kana inyo ang gamiton to process your samples. So, your closed system, kana siya actually ang isa sa disadvantage niya in terms of flexibility. Kumbaga, kung unsa namang good na siya ng mga hardware, software na nakabutang na daan dira sa inyuhang closed system analyzer, dili na ninyo siya mailisan. Not unless dito gihapon sa same na manufacturer, kanang wala mo inyuhang own na freedom na kunwari gusto ninyo na style ani sa inyuhang analyzer is ani pero since kato sa inyong manufacturer kay the other one so wala mo choice dili ninyo siya ma-change. But with closed system lang po, mao na siya ang advantage sa inyong open system kay since it comes from different manufacturers put together as one. So kung naamoy gusto i-change, dali lang po siya. With closed system analyzer, even ang reagents, dapat dito siya sa inyuhang manufacturer gihapon. Dito gihapon ninyo siya dapat i-purchase. And so far, sa mga machines, mga auto-analyzers na akong na-encounter during my work in the laboratory, usually mga closed systems yun. Closed system analyzers yun ang naa. Now, liquid reagents for open systems are less expensive than your closed analyzers. Closed systems contain a hidden cost advantage because reconstitution or preparation of your reagents for use does not require a technologist's time. So, yes, of course, since it is already prepared and manufactured or given to you by the manufacturers, all you need to do is just to place those reagents inside your machine. Unlike when we do in-house reagents, syempre ikaw pa mismo ang mag-prepare. So, mas laborious siya. So, that is why, as mentioned here, na hidden cost advantage yun inyong closed system. So, I hope nasabtan to siya. Now, in terms also of your reagent, naapot ay two types. We have your liquid reagent and your dry reagent. Syempre, understandable when we say liquid reagent, na meaning in a liquid form, and mao yun na siya ang kadalasan na inyo makita. But there are also reagent na kaning ginatawag nato na dry reagents. So, unsa ni siya? Most of your dry reagents are kumbaga na ay lyophilized powder sa sulod. It may be bottled as lyophilized na powder. When we say lyophilized powder, freeze-dried siya na powder. And of course, it requires reconstitution with either a water or a buffer. If nag-face-to-face ta karon, if naamoy clean chem na laboratory, you could actually encounter this too. Na naatay mga liquid reagents na ready to use na diretsyo na lang dayon ninyo mix dito sa inyong sample. Naapot ay mga reagents na dry na kung makita ninyo in powder form pa siya and you still need to place a water or buffer as mentioned here na para magamit ninyo siya. So again, dry reagent requires reconstitution. Now, one example or one type of dry reagent is your dry chemistry slide. Now, dry chemistry slide have microscopically thin layers of dry reagents mounted on a plastic support, and slides are approximately the size and thickness of a postage stamp. Now, this dry chemistry slide is actually an example of your thin analysis film. Diba? Remember the history na naatay thin film analysis technology. And your dry chemistry slide is already an example for that. So, i-present lang po na ako na siya later. Now, as you can see here in these images, basig matingala mo ma'am na alagining dipstick. This is actually an example of your dry reagent or kanang inyuhang not dili exactly kanang dry chem o dry chemistry slide or your dry reagent na in fact kaning kay wala pa man pud siguro wala pa man mo di ba nag urinalysis nag UBF so bale Each of this pad is already incorporated with the reagents necessary to identify what needs to be identified. And 'di ba, dry since dry, 'di ba? And all you need to do is just to dip, and then ang kaning mga colors na inyohang makita diri there will be changes, and then inyohan na siyang tan-awon dayon diri sa inyohang color chart to know kung unsa ang meaning ato na color changes. And then you will be able to know also the results na dayon sa inyohang patient. So again, that is the earliest and best example for that. Now the rest of this, these are actually your dry chemistry slides and an example of your thin film analysis technology. Diba? Diri pa lang, this is really the actual size of this. And this is actually very thin. Nipison good ni siya na tanan na naa sa sulod ani sa inyuhang template na tanan ninyong reagents na needed. Now, all you have to do is just to add your sample, and then leave it for how many minutes, and then i-read na dayon na siya sa inyong machine. It could be through the use of your spectro, commonly spectro. So, let us discuss now the different layers present in your dry chemistry slide, or what we could also known as your dry slide technology. Now, commonly there are just three layers. Ang common or ang basic good, there are just three layers here in your dry slide technology. We have your spreading layer, then your central layer, and your indicator layer. But as you can see here, in our image, there are actually a lot of layers. Na-mention nato si spreading, na-mention nato si indicator. So, unsa na itong mga central layers? Kanina siya, your scavenger layer and your reagent layer. And then sometimes, nagadungag pag yung taaning support layer. Now, the meaning of each of these layers or unsa ilang purpose is actually already written here in this image. For your spreading layer, this is where your sample is being distributed evenly. For your scavenger layer, this filters out substances that interferes with your results. Then for your reagent layer, from the name itself, reagent, so that is where your reagent will react with your sample, that is where your reagent is placed. Then you have your indicator layer, reacted sample collects for spectral analysis. So dara na siya. And then support layer is for your optical interference. Na kumbaga, mura siyang mag-serve as monochromator na siya ang magdara ayan, dili day monochromator. It could actually serve as your exit slit na para dili makasulod ang other na wavelengths or naka wavelength of light. So that is the purpose of your support layer. And then as mentioned, you just give time after adding your sample, just give time for it to really proceed the whole process, and then spectrum na dayon ang last. Dara nato na siya i-read. Okay, diba? As you all know, spectrophotometer, color development man jud na, mga colors man jud na ang iyahang gina-read, which is also what is being done here in your dry slide technology, color development. So, dry chemistry slide, in your reagent layer, it might contain enzymes, dye precursor, and buffers necessary for the analysis of a specific component. Sample control or standard is deposited on your spreading layer, and then selected components are allowed to penetrate to the reaction layer, which in turn activate your dehydrated reagents. So, nga na naka-nice ang dry chemistry slide. Diba? No need for you to mix reagent, na magmano-mano pa. Very simple and less time consuming and less labor use. I forgot also to emphasize that between your liquid and dry reagent, it would always be best to have a dry reagent. Why? Number one, mas longer iyahang kasi tawagan mas dugay siya ma-preserve. Kay kung reconstituted naman gud siya daan or inyuhang liquid reagent, mas dali siya madaot. Unlike sa inyuhang manggong dry reagent na since naka-powder form pa man siya, wala pa ninyo gigamit, so mas longer iyahang shelf life. So, panata siya. I forgot to point out that one. So this video would present to you your dry chemistry slide as to unsa ang whole na process. So kindly watch this video.
Running in-house laboratory diagnostics allows the advantage of instantaneous medical results and rapid initiation of treatment. This compact machine analyzes up to 31 clinical chemistry parameters. Using the plasma filter reduces turnaround time by eliminating the centrifugation step and increases patient safety by avoiding handling errors. The high degree of automation allows even non-specialists to operate the analyzer. Only 3 steps to accurate analysis. Simply set the sample and the plasma filter, set the ready-to-use slide, and press start. The plasma filter prepares the sample for analyzing within 1 minute. Alternatively, laboratory staff can use a centrifuge and no plasma filter is required. The choice is yours. Up to 20 parameter slides can be processed simultaneously in one process cycle. Each parameter requires just a few microliters of serum or plasma. Analysis results can be easily transferred to your laboratory information system via the computer interface. The DryChem NX500 quickly provides you with precise laboratory results, even at night and on weekends, with no setup time. Storage of the parameter slides occurs at a temperature range of +2 to 8 degrees Celsius. Swipe the QC card just once per enzyme or substrate to receive accurate measurement results for each slide lot. Coding allows the slides to be read in random order. If necessary, the analyzer dilutes specimens automatically to a preset dilution ratio and displays the converted end result correctly. Our FDC analyzers are the market leader in Japan and have been used worldwide since 2006. Accurate and reliable results are verified by external laboratory tests.
O, di ba? Hope you appreciate it, kids. And actually, I am also shocked in terms of the brand Fujifilm. Kasi di ba we all know that Fujifilm is just a more on sa photography, camera, and I was actually amazed na somehow naapo din ni sila yung mga yung ani na products. So, what are the techniques now of preservation in terms of your reagent? So, keep all reagents refrigerated. Kani man gyud ni siya ang, 'di ba, common way gyud of preserving and prolonging the shelf life of your different items magyud labi na if reagent kung liquid na siya or food. So keep all reagents refrigerated until the moment of need and then quickly pre-incubate them to reaction temperature or store them in a refrigerated compartment on the analyzer that feeds directly to the dispensing area. There are some reagents, and actually kani gyud ni siya ang common na mistake sa mga students or interns kay kani laging nagadali-dali. Even ako personally ha, during my work in the laboratory na there are times yun na makalimot na ko ani na part na by the time that you remove your reagents in the refrigerator, do not immediately use it. As mentioned here, 'di ba, pre-incubate them or you need to ano sa kanang dili kanagong pawalaon sa nimo ang kabugnaw sa imuhang sample, sa imuhang reagent rather, kay temperature could actually affect the processing of your results. Then provide reagents in a dry tablet form and reconstitute them when the test is to be run. So kanina, I think that I emphasized this well. Other than your lyophilized na powder, there are times na naka-tablet form siya ang inyuhang reagent na then again, either way where na naka-lyophilized ba siya na powder or naka-tablet form, same process, you still need reconstitution for that. And then manufacture the reagent in two stable components that will be combined at the moment of reaction. Ang ginamin lang man ani kids, instead of combining immediately your reagents in one bottle, better na i-separate sa siya and then kanang kamo na mismo ang mixing kay para mas longer iyang shelf life, mas dugay siya mag-expire. Then, reagent delivery techniques. This is in terms of sa machine na ninyo mismo as to how it will be introduced to your sample or how it will get reagent. So it could either be through syringes driven by a stepping motor, pipette the reagents into your reaction container. Or it could also be through piston-driven pumps connected by tubing, which may also dispense reagents. Or it could be with the use of pressurized reagent bottles connected by tubing to dispensing valves. Now, these pressurized reagent bottles are computer controlled na kumbaga in terms of the opening and closing of your valves, computer na ang mag-control as to when sila magkuha or dili sa inyuhang reagents. The fill volume of reagent into the reaction container is determined by the precise amount of time the valve remains open. So now we go to the next step, your chemical reaction phase. Now, your chemical reaction phase involves consists of four parts. So one, you have your mixing. Mixing could either be through forceful dispensing, magnetic stirring, vigorous lateral displacement, rotating paddle, or ultrasonic energy. Now, regardless of what step, actually, ang isa sa pinakalisod yun i-automate is kaning mixing na part. Naay uban na machine kay naman jud uban machine good na grabe ka transparent na makita gyud ninyo ang overall na process. There are some machines na ang style sa mixing is mura lang nilang i-vibrate, something like that. But then again, itong na-mention ako about your hematology analyzers na literal na mixing siya, nakatong usahay mura siya loop of 8 ninyo or usahay upside down na style. So, kana ang ginabuhat pod. In terms of kaning lateral displacement, actually with your discrete analyzer, it is positive displacement na type. And then, I guess you are familiar with magnetic stirring. Usually, ginabuhat man ni siya sa mga chemistry. Labi na sa sige ito na subject. Basta na ato yung isa ka certain na subject. Basta again, chemistry na butang mo og magnetic stir, di ba? And then siya na ang mag-stir. Wala na siya inyuhang different types of mixing. Then, separation. Separation in relation to in relation to removing interferences. Mao man ni siyang ginamit sa separation here. Separating your desired substance to the other substances not needed and interferences. So to separate properly, you need to use a very high reagent to sample ratio. In terms of this, meaning since taas man reagent to sample ratio, meaning grabe ka diluted ang inyuhang sample. And ang reason daw for that, nga no i-dilute ninyo og tama ng sample so that any turbidity caused daw by your precipitated protein, kay dili daw siya ma-sense sa inyuhang spectrum. And then, pwede po i-shorten ang reaction time to eliminate slower reacting interferences. So next step is incubation. Most adiliday most. There are cases na ang inyuhang mga testing processes dapat gyud siya naka 37 degrees Celsius. Pero mas daghan man gyud ang naka room temp lang, but if it needs to be incubated properly, then a heating bath will be needed. So nanay naka-incorporate na daan sa inyong machine na heating bath which will maintain the required temperature of your reaction mixture. And another purpose of your heating bath is to provide the delay necessary to allow complete color development of your sample to your reagent. So the components of your heating bath are the following: your heat transfer medium, your heating element, and your thermoregulator. Then, we have your reaction time. When we say reaction time, it is the completion of your reaction. So it is the rate at which the reaction is proceeding. And isa man sa ato ang way of calculating actually kung pila ang reaction time, calculating the amount or the level of your certain substances in our body. So we need to get the reaction time. Next is your measurement phase. Now, here in the measurement phase, balik lang ta dito sa different na kanang methods na na-discuss sa inyo ha, sa katong instrumentation ninyo na part, that it could either be UV light, fluorescence, diba, flame photometry, ion selective electrode, gamma counters, luminometers. These are different methods that can be used to measure your product. Depende lang yun na siya sa kung unsa na ni na product or na substance ang ato ang gusto na i-measure na kung asa ang best pud na gamiton na method. But the most common method still is your visible and UV spectrophotometer. Analyzers that measure light need monochromators. So you already know the purpose of your monochromator to produce specific wavelength. Then classically filter wheels have been used to separate light, which are usually computer controlled. Next, now we go to your signal processing and data handling. Now, accurate calibration is essential to obtain reliable results. Now, what is this calibration? When we do calibration, this is a way of helping mura maning assurance gud sa ato na accurate yun ni ang measurements. And calibration is done through comparing a known na measurement, which is your standard, to a measurement uh, through the measurement being used in your instrument. So, dara po na musulod ang inyong mga controls. So, as mentioned here, diba, this requires proper use of your standard, which then reflect the data on the standard curve according to which the sample results are interpreted. Recall what are the standards. When we say standard solution, it is a solution containing a precisely precisely known concentration of a substance. And kana na siya ang ginagamit nato for calibration. Sa tanan siguro section sa laboratory, medyo hago gyud ang starting. Ang starting sa inyuhang chem. Kaya you need to always run certain, dili man po jud everyday ang calibration ha, but you need to always run controls. And if by the first time nag-run ninyo ang control, dili siya mopasok, then that is a problem. Kaya you need not proceed your processing of samples unless ma-okay ang inyuhang controls. Kaya kung ipadayon gani gihapon na ninyo, I tell you, tanan-tanan results na mugawas dira mali. So, kana lang. Mo na ang gina-term na mo before na toxic dara sa chem na kung dili gani mosulod ang controls, dugay kay mosulod, and then daghan nag naka-standby na samples. So wala na, mura na kag mabuang kay syempre in clinical chemistry si chem manggud jud na ang grabe gyud na puro machines gyud na inyong kaatubang, and kung kana sila na mga machine puro guba, hilak na lang gyud. ingana gyud ang mahitabo. So after the calibration has been performed and the chemical or electrical analysis of the specimen is either in progress or completed, the instrument's computer goes into data acquisition and calculation mode. So this process may involve signal averaging, which may involve hundred of data pulses per second. So, in terms of this calculation mode, data acquisition, actually I think diri mosulod yun sa signal processing and data handling po ninyo ang kanang QA and QC, quality control and quality assurance. During my work in the laboratory, dira pag yun na ako totally na appreciate yun ng inyuhang Levy Jennings chart. Kay I think na-mention naman po din ako sa inyo ha, sa labi na katong mga na-under sa aqua in PMLS siguro, na before I was actually handling your G6PD confirmatory testing, na 'di ba akong usual na patients kay dili mga teenage, dili mga adult, mga babies, mga newborns pag yun. And in the G6PD confirmatory testing mga good kids, isa sa amo ang responsibility is to really kana in-note yun nako always dapat ako ang control values, and then every end of the month kailangan magud mag-submit na dira ang kanang Levy Jennings na dapat mo create ko og Levy Jennings chart. But it's a good thing na sa syempre dili naman dili naman ta mubalik atong manual, naman pud na sila software na dito na lang nako i-input, and then dira na ko jud makita na katong inyong mga gina-mention na hala kanina day out sa control, mga ing-ana. So dara na nako na siya na-appreciate. Grabe ka trabahoso gyud ang D6PD confirmatory testing gyud sa katong ako na time kay kumbaga murag naa pa sila sa process manggod of nakanang ginahan-ay pa siya na wala pa'y tarong na software, wala pa'y tarong na mga kuan. Anyway, nagsige na kog chika, so nitaas na yung time. You may skip that part. So, we are done with the different steps in your automated analysis. So again, kindly just read this table. Could give you additional inputs about the different steps. But just to add also, kids, these are also automations done in the pre-analytic and post-analytic steps. Although some of these have already been mentioned, especially about, diba? Especially about decapping, aliquoting, recapping. Let me just point out also LIS receiving. This is actually a part of automation and actually grabe good kanindot unta ning LIS or labi na kumayon ta og HIS good. And I think you already know that kay di ba nanabiya jud mo yung HIS na ang nakanindot man gud anang naay hospital information system. Labi na kung kanang sa labi na sa kanang in terms of receiving of requests kay dili ka minimal ang errors labi na sa kanang ma-mislook ang request mga ana and then even ang pag send sa mga result dili na necessary na ikaw ang mohatod dito sa nurse station. Everything is already programmed in the computer na all you have to do is just click some and then search some and then makita na dayon na siya tanan, which is actually very good kay diba, and dapat environment friendly punta dili na takala sa papel. So kana siya. So now, for the last part of our discussion, what are the future trends now in automation? So automation will continue to evolve, yes. System integration and miniaturation with more technologically advanced computer power will persist to accommodate more portable analyzers for more precise testing. This is actually true, especially kanina part na miniaturization na kumbaga, if the machines before dinagkuay, as automation evolve, the more po na murag nagagamay po na ang inyuhang mga auto-analyzers pero mas bongga ang iyahang system. And of course, the reason for that is so that dili dako po niyahang space na makuha sa lab. Kana man gyud na siya. And as of the moment, daghan na kaayo og POCT, which is also a trend in your automation. Na as much as possible ang ato mga patients kung pwede gani pud ang kaning mga machines na for testing kay naa sa ilahang balay and then pwede gani na sila na lang ang mag-process so kana di ba your POCT, your point of care testing. So that is one example also of the future trends here in automation na dili na nila need mo adto pa og hospital to travel na nadayon sa ilahang house. Automated analyzers will have artificial intelligence whereby the computer will think or make decisions if sufficiently programmed with infinite scenarios of data. So, di ba, amazing kaayo na ang computer na po mismo mag-think. Siya na mismo mag-ingon na. Actually, naatay mga machines like for example sa inyuhang AUBF sa inyong urinalysis naman jud tay machine for that na ginapresent pa hinuon niya sa ato kung unsa nang naay mga errors na kailangan ninyo i-view ang inyuha yung urine sample sa microscope na po na siya na mga ingana na processes. And then spectral mapping or multiple wavelength monitoring with high resolution photometers and polychromators will become a standard in your automation. O, di ba grabe, unedited kaayo ang ako ang video. So anyway, that actually ends our discussion. So one last video na lang yun to present kids just so you can have a overall na view lang po in terms of your auto analyzers. Okay?
The BS480 with an all-new operation software interface offers 400 tests per hour for general chemistries and 560 tests per hour throughput, including ISE testing. The reagent carousel provides 24-hour non-stop cooling at 2 to 10 degrees. It stores up to 80 reagent bottles, including 2 utility reagent slots for DI water and wash solution. The sample carousel contains 90 positions and accommodates primary sample tubes, an aliquote tube, or micro-insert cups. The sample carousel is made with conductive material which greatly improves the sensitivity of the liquid level detection system. The BS480 uses reusable glass cuvettes in the reaction carousel. All 90 permanent cuvettes are automatically cleaned by the 8-step auto wash station. The probes are cleaned internally by high water pressure and externally by the latest waterfall design, ensuring thorough cleaning. Should there be a need for deep cleaning, an enhanced washing function can be requested in the software. This laboratory workhorse is equipped with 2 separate sets of probes and mixers to be used with the reagent and sample carousel. Internal barcode reader enhances sample and reagent management, ensuring that the right test is performed on the right patient. The BS480 is highly compatible with major LIS systems, providing bidirectional interface transmission. The optical system utilizes grating photometry and a halogen tungsten lamp. 12 wavelengths from 340 nanometers to 800 nanometers are available to accommodate most clinical chemistry testing requirements. The convenient front-load design allows easy access to the liquid bottles and the all-in-one ISE reagent pack. The ISE module consists of a sodium, potassium, and chloride electrode and is located on the side panel for easy access. There are 2 independent power switches. The main power switch controls reagent refrigeration and maintains the overall system temperature. The analyzer power switch controls the mechanical parts, provides energy savings, and prolongs lamp life while the instrument is not being used during the night and/or weekend.
So, I hope you appreciate the video. 'Di ba? So na-mention didto, 'di ba, si throughput, na-mention ang katong inyuha mga wash solution, katong DI na yang gi-mention, that is your deionized no water. Na-mention si ISC, that is your ion selective electrode, which is usually the method used for your electrolytes, 'di ba? Sila sodium chloride, unsa pa ba to ang na-mention, or katong naay cooling system, si LIS. So mo na nagipili gyud nako ni na video kay at least medyo na-mention gyud niya ang katong ato ang mga na-discuss diri sa ato ang topic.